wrapping up chapter 20, talking about the rest of the plant parts and then how water and minerals as well as carbohydrates actually move through the plant. All right, now moving into our leaves. The leaves are usually the most prominent organ that we kind of think about. So their growth comes from what is referred to as marginal meristems, and they grow outwardly and ultimately form that blade of the leaf, kind of the flat portion. And then once the leaf's fully expanded, that marginal meristem actually stops. So the leaf doesn't continue getting bigger throughout the existence of it. It gets to its size fully formed, and then it stops growing. So again, our leaf blade would be here, just that flattened region. Of course, they don't all look the same, especially when we think about the pine needles. Those blades look much different than like a maple tree or our poinsettia leaves that we see here too. All right, so some of the things that we see on the leaf, there are a few different parts. Again, that stalk where it actually attaches into the stem, that's called the petiole. The two leaf-like organs that start there as it first starts to mature, those are called stipules. So they kind of flank the petiole. Then it has leaf veins. Again, that's vascular, so it has their xylem and phloem. Now, in most dicots, those veins have kind of a net or reticulate venation, just meaning they branch out. Whereas in monocots, those veins form a parallel structure. We can see that here again, our dicot here on the left, just again, lots of branching that we see of those veins versus this parallel arrangement that we see in the monocots. Now the leaf blade has a couple different forms. There can just be a simple leaf, which as it implies is just a single undivided blade. But there are also compound leaves that have multiple blades called leaflets. Pinnately compound describes those leaflets that are arranged in pairs and they have a central axis. And then the palmately compound, those are the leaflets that radiate out from a common point at the end of that petiole. So a little different arrangement that we can see. So the different patterns, we can have alternate, and this is where the leaves spiral around the shoot. We see this with corn. We have opposite, where they occur on the opposite side of the shoot, and then the whorled, which they kind of circle the stem as a group. And just kind of seeing these alternate, this is kind of what we think about with most plants, but again, they alternate. So we have one here on the right, so the next one's go on the left, and then the next one's on the right, the next one be on the left, and right and left, right and left. Opposite periwinkle, so they're both coming out, and then one goes to one side and one goes to the direct opposite. And then whorled would be they come out and they form essentially a circle of blades around that one stem or that one petiole. All right, now looking at our mesophyll. So a typical leaf has a massive parenchyma, again, those actively functioning, going through photosynthesis cells that make up what's called mesophyll. And that's where the vascular bundles or veins are actually going to run. So it's loosely packed column-like layers of those parenchyma cells are underneath the upper epidermis of the leaf. And that's referred to as palisade mesophyll. We'll see a picture here in a second that has some packed chloroplasts, but then the rest of the leaf has what's referred to as spongy mesophyll, and there it has lots of interior spaces that allow for gas exchange. So essentially, if they're closely packed, we call that palisade, so packed palisade. If there are a lot of holes and it looks more like a sponge, then we have spongy mesophyll. Again, just kind of looking at the difference here. So both of these have that mesophyll. It's just here with those cells tightly packed together. That's our palisade. Whereas down toward the bottom, we have more loosely packed cells, and that makes up the spongy mesophyll. And again, the idea here is that there's more room for gas exchange to occur, especially close to those stoma. And now just focusing on how water actually moves through the plant. So of course the vascular plants have that transporting using the xylem in the phloem. And when we're talking about water, of course, the xylem. But those water and minerals enter through the roots, so through those root hairs, and then they're transported in the xylem. Now the carbohydrates, again, the food, from photosynthesis, again, they're transported by the phloem. So again, xylem, water, phloem, food. So just kind of the flow of materials into, out of, and within. So this is, again, just showing the phloem here. That's going to be the carbohydrates. Again, the xylem kind of here in the pink, that's going to be the water and the minerals moving through those. And then just the picture here looking at the whole plant. So again, water and minerals coming in through the root hairs, into that going through the xylem up to the shoot system, and then to the leaves for photosynthesis. They're going to exit through the stomata, which is what is showing here. So here's a stoma. Again, that water vapor, whenever they're opened and those guard cells are there opening the stoma, then the water exits through that way. So again, it enters through the roots and it exits through the stomata. And to get from point A to B is through that xylem. Now, as water's moving up, there are several factors that help the water move up, because of course that's going against gravity. So that initial movement into the roots involves osmosis, which is the movement of water based on the concentration. So like hypotonic, hypertonic. So since there are more ions and more nutrients inside the cells than there is in the soil, that pure water from the soil moves into the hypertonic solution. So it moves into the cells, again, because the fluid in the xylem has more solutes. Again, hypertonic is the term that we use for that. So the inside of the cell is hypertonic. So that osmotic force creates root pressure, and that causes water to rush in to those hypertonic cells, and then it pushes the water up the plant stem. Now it also uses what's called capillary action, and you can see this if you've ever taken a straw in a drink and you put your finger over the one end of the straw and you take it out. Of course, whatever your drink is stays inside of the straw. That's capillary action. Or if you dip a paper towel into a fluid, it kind of runs up the paper towel. All right, so again, this capillary action essentially pulls on the water and moves it up the plant. So essentially it results from tiny electrical attractions between the water molecules that are polar, meaning they have a little bit of charge, to the surfaces that carry that electric charge. The term also goes along with adhesion, where water is attracted to other polar molecules, like the cell. So as it, again, that water on the paper towel, it moves up the paper towel by this adhesion or capillary action. But by itself, though, it is not strong enough to pull water up the entire plant stem. And this is just showing, again, the example of the straw. So again, if you just place a straw inside of a beaker of water or in solution, again, if you would like put your finger over the top here and then pull it out, that liquid is going to remain in there. Again, that's adhesion. It's attracted to the polar molecules of the glass or whatever the straw is made up of. This is also showing that the smaller the diameter, like this little one here, that there's more attraction. It can pull the water up farther than a wider diameter. So that goes in with those vascular bundles. They are thin, small diameters. That allows the water to move farther up the stem than if it had wide cells where less water would be pulled up. So it loses that pressure. Now another term, cohesion. So we have capillary action and adhesion. But in addition, cohesion is kind of this final pull of the process, is moved by what's called transpiration. So water actually evaporates from the top, so the leaf of the tube, and that's going to pull the water up from the bottom. So essentially, the escape of water and the evaporation of water pulls the water from further down the stem or into the roots up that. And the column of water doesn't collapse because, again, that water is attracted together, and that attraction of water to water is called cohesion. So if it's water to another polar molecule besides water, it's adhesion, again, like paper towel or another cell, but water to water is cohesion. And again, as the picture just showed, the narrower the diameter of the tube, the more tensile strength, so it allows more movement up that water column. All right, so then we have this adhesion, cohesion, tension theory. So the combination of gravity, that tensile strength, and cohesion as it's attracted together is what explains the movement of water up through that chute system. So it's referred to as the cohesion, adhesion, tension theory. Now, as we mentioned, transpiration, which is how the water is leaving the plant. So about 90%, so the majority of water that comes in actually gets lost in the atmosphere. And most of that is occurring through the leaves, and it's going to be moving out. So first, though, it passes through those pockets of air and that spongy mesophyll. Again, palisade is the packed, spongy has more for the gas exchange. And then it ultimately evaporates through the stoma. So with high humidity and low temperatures, that increases the transpiration rate because those stomas stay open and it pulls the water out into the atmosphere. And this is just showing that process. So again, if there is kind of this dry air, it's going to pass. So that dry air passes across the leaves, causes that water vapor to evaporate out of those stomata. So it's pulling it from where there's a lot of water in the leaves to where there's less water, so in the air. So that loss of water from the leaves essentially acts as a suction and draws water up from the roots. So now newer water is going to enter the plant through those roots to replace the water that was lost by the stem. So again, the more water that transpires, that evaporates, that's going to pull more water in from the roots into the shoot system. So plants need to be able to deal with their water loss. So the only way they can really control that water loss kind of on short term is to close their stoma or the stomata. However, they have to balance that closing to prevent water loss with the closing, which then prevents access to carbon dioxide. So they're open to allow gas exchange. So if they close them to keep the water in, now they have the problem of not having carbon dioxide for photosynthesis. And actually what causes those stoma to open or changes in the water pressure of those guard cells that surround them. So as we see here with the guard cells, so the guard cells are plump and swollen with water. That creates turgor pressure. So the cells are said to be turgid. And whenever those guard cells, shown here in green, when they're nice and full, kind of and plump, then that opens the stoma. So that's going to allow the gas exchange, which means water is leaving through that as well. So then whenever enough water transpires out of them, then those guard cells lose that water, and it causes them to kind of collapse, and the stoma is actually going to close, which is what we see here. So again, whenever they're nice and plump, they fill up, it pushes them apart, which opens the stoma. Whenever they become kind of more wilted due to water leaving, they collapse on each other and it closes the stoma. Talking about the water, of course, the root hairs are very important because that's where the water ultimately comes into the plant. And again, as we mentioned, that increases the surface area, just means more water can come in. So back to turgid, those root hairs are also turgid, meaning they're kind of plump and full of water because they contain, again, that higher concentration of solutes. That's called hypertonic. So they are hypertonic, which means water is flooding in or coming in trying to dilute out all those solutes. They also have different ion transport channels, so that is going to allow the transport of specific ions to keep a hypertonic environment. And some of that may involve active transport. So energy may need to be expended to keep those cells hypertonic, which allows water to keep coming into them. And then those minerals are transported by the xylem because they're dissolved against solutes inside of the water. And this is just, again, showing those root hairs. So the more root hairs there are, the more water and minerals that can ultimately be absorbed. Now, as far as carbohydrate transport, so again, now we're talking about the food or phloem. So this transport is called translocation, just changing location. This is mainly how different carbohydrates are moved, again, through the phloem. So xylem is for water, phloem for food. It uses a process called mass flow, which is passive, and it occurs due to water pressure. Now, when essentially the carbohydrates are loaded into those sieve tubes that make up the phloem, water also comes in during osmosis. Again, water essentially goes with solutes like glucose or ions. So that water pressure then forces the carbohydrate down the plant. So an area where sucrose, which is sugar, is made is called a source, which would be like our leaves. So that's the source where the carbohydrate is made. And then where it is delivered from that sieve tube is referred to as a sink. So where it gets stored is the sink. So where it's made, source, where it's stored, sink. So it moves from a source to a sink by this, what's referred to as the pressure flow hypothesis, which is that it's moving with the water as that pressure is changing. So here, this is showing this translocation. So again, our sugars created in the leaves here by photosynthesis. So that's our source. Here is the leaf cell, those parenchyma cells. It enters by active transport. So the way that it gets into that phloem is using energy. Now, once it's in there, though, again, now we have that hypertonic condition, so that sugar concentration increases, and that draws water into the phloem cells. And that's through a process of osmosis. So that doesn't need any energy. The addition of the water in with that carbohydrate, again, that creates that water pressure to build up. And once it builds up, it pushes that sugar and those carbohydrates down through the shoot and into the roots. And then ultimately, they enter those root cells, which is the sink. They need active transport for that. So the flowing down is by water and osmosis. That doesn't need energy. But getting the sugar into the phloem and out of the phloem does require some energy.